and welcome to the lecture focusing on quality by design concept. My name is Jukka Rantanen, and I will, during this coming 45 minutes, introduce the topic quality by design and bring some scientific perspective to this expression. So quality by design, it can be also understood as a regulatory term, but there's quite a lot of science behind it, and that's something that we are now planning to walk through. And what I hope that we manage to walk through during this lecture is to give you some introduction and background for the quality by design approach. And I use this abbreviation QPD, that is commonly used expression for the topic. I will introduce very shortly the regulatory framework and then all where this is based on. And then most importantly, focus on quality by design toolbox. So the elements of QPD that you will need to implement this in practice. So quality by design work, it starts with a risk assessment of a given product. And then this product can be whatsoever. It can be a simple immediate release tablet or very complex, long-lasting injectable nano suspension. So it can be many different type of products that you can develop based on QPD principles. And risk assessment is the starting point for identifying the areas of the production or the product design that deserve more attention. And when you identify which part of your product are the most, so to say, risky ones, then you can direct your experimental activities on those ones. And that would be the base starting point for the DOE activities, design of experiments. And it is also important that you link these activities towards process analytical technologies, process measurements. So PAT is a set of different analytical tools that can be implemented into production environment. And by this means, we can increase the level of understanding of things what are happening during the processing, and then keeping the product under production under control. So the key goal of quality by design is that to build up a control strategy that helps you to keep the production and product within the desired specification. And as mentioned, there's a regulatory framework that is behind all these concepts. So this is all described quite well in the ICH International Conference on Harmonization documents, and especially the ICH Q8 document, Pharmaceutical Development, that is one of the key regulatory documents defining this concept. So all this you can find with a simple Google search. So if you Google ICH Q8, then you will find 60 pages of this document. I'm not planning to walk through it here with you, but if you one day need this document, then it's out there for you. So what do we mean with this QPD approach? So when you start developing pharmaceutical product, it can be, again, it can be solid product, it can be cream, it can be injectable. So you start with your raw materials, and the raw materials define your input properties, input space, so to say, mathematically. And in the next phase, you need a process, some processing solution that transforms this material into the final product. So in this case, we are making a simple immediate release product. But when you deal with pharmaceuticals, then there are always like surprises. So some of them are something that you do on purpose. So you can do intentional variation into this process environment. So you can change process parameters, and then by this means achieve what you were planning to do. But then there's also something that you don't know. So then there's unintentional variation. And the idea of quality by design is that you get an overview of all these sources of variation, how they affect the quality of your product. And then this you can do by doing careful risk analysis and design experiments. And if we think how, maybe not anymore, but in olden times, many old products, they were developed very traditionally. So even this expression trial and error has been used to describe the development process of some of the older products. And trial and error is of course something that we should not be doing. So we should base our product design on a structured experimental work. And another aspect of pharmaceutical business area is the relatively rigid structure of the quality system. So if you think about how we are releasing, for example, a batch of products, so all product release is based on testing. So we take samples from the end product, coated tablet, for example, and then as long as that product is within the specification, we measure 30 tablets out of 30 million tablets, and then everybody is happy with this type of approach. So this testing as a philosophy is quite deep in the pharmaceutical business area. And this quality by testing, it is a bit annoying aspect. So it is like a driving with a fixed steering wheel. So you can imagine that you are in the US driving 100 kilometers on a road that is just straight road. So everything is fine if you have no surprises. But the one day when you are seeing some surprise, so all of a sudden the road is doing something unexpected, then you are in difficulties. So what we need in the production of pharmaceutical systems is more flexibility, and then solutions that allow for changing things while you are driving, so to say. So you need to be able to change the direction of the process if things are not proceeding as you were expecting them to do. And when it comes to this trial and error, so how you should design your products is not always exactly like after the cooking book, but not your kitchen should not be looking like this. So you indeed should build up the product design principles on understanding of your product. So you need to first explore the materials and material properties, and then create understanding how variation affects your product quality. And this is the sole purpose of the quality by design approach. And if we think about how this quality by testing might look like, I have here a few graphical presentations of these expressions from FDA senior officer, Lawrence Yu. So he is head of one of the departments at the US FDA. And then he has been drawing this picture in pharmaceutical research a while ago. So in quality by testing thinking, we are having materials that are meeting the specification. And while we are processing, we always use fixed process parameters. And we might have some in-process control, but it is based on sampling and offline analysis of these samples. So often it's very difficult to change something if you are seeing problems. So if you fail these in-process controls, then you basically have to discard the whole batch. And that is expensive and definitely not wanted scenario. And the most of the testing is on the end product. So then you do lots of end product testing and uniformity, dissolution, residual solvent, all possible type of measurements. And then if you are failing in these, then you need to discard again the whole batch. So this is very inefficient and not the ideal way of producing. So if we contrast this scenario here, so this is something that we can call quality by testing. So if we would like to go with quality by design approaches, that we allow flexibility in these process parameters. So we have process control routines that allow you to change process properties during process parameters during processing. So for example, if your granules are not drying in the fluid bed drying process fast enough, it seems that there's something strange happening. So then you are allowed to increase the temperature. So there's flexibility in these process parameters. And that is the sole purpose of the quality by design approach. So the thinking that you generate first enough of process understanding, and then you understand where it is safe to operate. And then you allow the process more freedom to be changed if needed. And by this means, then when you know that your material is within the specification already during processing, then you can reduce the end product testing. So you don't need that many specifications for the end product and then that heavy testing anymore. So acceptance criteria can be based on performance, and then part of the testing can be reduced, and then it can be even the product can be fully based on real-time release testing, and that's an important concept when it comes to process analytics. So you collect enough information during processing so that you can release the batch immediately after production, and that would be efficient and then the cost-saving approach for the production. So if we contrast these two scenarios with this visual expression, so here we are having again the input properties and then processing level and then output quality of your product. So if you have a variation in the input properties like a quality of an excipient, if you're using fixed process conditions, then you just simply transfer that variation into the quality of your product. If you then, on the other hand, build up process measurements, process understanding, and then control loops in the process, so then you can change process properties. So when you are seeing this variation here, so you go from this to here. So for example, the moisture content of your excipient is changing from high to very low. Then you can change something in the process so that you always get the desired product quality. So this is simply the purpose and sole purpose of a quality by design approach. And in this context, then these expressions are important. So you need to understand the design space around your product. So design space is referring to this variation, the known variation, and then knowing how it affects the quality of the product. And then control strategy is a strategy that you can cope with this variation. Process analytical technologies, PAT, is referring to a group of different techniques that can be used for measuring things from process. So with PAT, we are referring to analytical techniques that can measure moving material. So instead of taking a sample and dissolving it to solvent and doing HPLC of that, then what we would like to do is to do fast measurements for moving material, even without any sampling or sample preparation. So that would be ideal, and it can happen in seconds instead of spending hours in HPLC analysis. And then these control loops are, so to say, engineering expressions for changing the process automatically. So based on the measurement, then you can change something in the process that affects that property that is being measured. So for example, if you are measuring the moisture content of granules, then you can automatically change the heating temperature so that you can get the desired drying rate for the granules. So this would be an example of a control loop. And here is again the toolbox that is needed for creating this process understanding and doing the quality by design approach, or if you will, risk-based product development. So this expression risk-based is also referring to the, so to say, full-blown quality by design approach. So risk-based means that you understand the risk related to your product, and then that is done by doing risk assessment of your product. And after risk assessment, then you do your structured experimental work. You do this DOE, design of experiments, and that gives you understanding how variation in the process affects the product quality. And in many cases, you need to do material science and understand the material properties in very detail, very fine detail. So basically, you need to understand the molecular level phenomena so that you can explain why things are happening as they appear. So risk assessment and then DOE, they are typically providing you like a correlation. So if I'm changing temperature, then the granule granule moisture content is decreasing. So that is quite obvious, but you can find these mathematical correlations. But then in order to understand what is really happening in there, then you might need more complex analytical tools for explaining this phenomena. And in order to keep things during processing under control, so then you would need these process analytical technology, PAT tools. Yes, so I think this was basically the first part of the lecture, and I can see that we have a high number of participants here, so that's nice. Maybe one of you has a question here that we could take a short discussion here with this first part of the lecture. If there's a brave individual asking something. Seems very silent, so maybe I'm not grilling you more, and then we just simply proceed. So in the next set of slides, I would like to walk through a little bit more about this process analytics. So what I'm meaning with the process analytical technologies. And as I mentioned, process analytics or process analysis is referring to techniques that can be used for measuring pharmaceutical materials during production. So in an ideal case, these measurement techniques allow for integration with the process stream, the moving material, without disturbing the material flow. And here I have collected a few examples of process analysis. So this example is related to small molecule product development. So we start with a synthesis of an API, and we recrystallize that, and then we might need granulation mixing processes before proceeding to tabletting and finally to coating of the tablets. So this is complex series of processing steps, and if something starts going wrong in the crystallization process, it might be something that is then escalating in its processing step. So if you have, for example, a polymorphic impurity in crystallization, and then you increase amount of that in the granulation process, and then by compaction, you induce some sort of degradation process, and then you coat it, and then after coating, you do sampling and then find out that there was something, there's too much of degradation product in there. So then it's very, very inefficient. So you might have spent already 2 weeks on this processing and then several million euros on this production. And then you should have seen it already in the crystallization step. So then the ideal solution would be to control and measure the crystallization process so that you understand what is happening in there. And this can be done with the different process analytical tools. And then these spectroscopic tools, infrared, near-infrared, Raman, terahertz, they allow for direct measurements from moving material. And things like polymorphic impurities can be identified with many of these spectroscopic tools during crystallization. We can do image analysis from moving material. We can do laser diffraction, X-ray diffraction methods for looking into material properties during processing. And these techniques allow for very precise particle size measurement, and even polymorphism-related phenomena can be measured real time. Tabletting process can be measured with by looking into mechanical properties and thermal, even acoustic information, and electrostatic charging of material can be measured directly from the moving material. So there's quite a lot of different process analytical tools out there, and I would like to in this lecture walk through a few examples so that you get an idea of what these potentially could be used for. So if we take these processing steps here and start thinking about what you can do with spectroscopy. So one of the most commonly used spectroscopic tools is a near-infrared technology, NIR spectroscopy, that is used in many steps of this process train here. So we can use NIR for identification of material. We can measure water content, hydrates, polymorphs can be identified with NIR. And it is also commonly used solution for following powder mixing processes. So powder mixing is a difficult process. So when have you been mixing enough before tabletting? So NIR can give you a direct measurement of the degree of mixing in the process. We can also measure amount of active in the tablets. So, and then since this can be done in a millisecond scale, so we can basically have a quantitative measurement of tablet amount of active in tablets of 100,000, 500,000 tablets. So you can really analyze a high number of dosage units, and that's the beauty of these spectroscopies. And coating layer thicknesses is then one of the parameters that can be measured here at the end of the production cycle. So you can see that with the same analytical tool, we can measure basically from each processing step some relevant piece of information. And if in your specific product, if it seems that the water content is critical for the quality, so then it could be worth investing NIR spectrometer for looking into drying process or granulation process. We can pick up another analytical tool, and then, for example, in infrared measurements can be also done in a process mode. So infrared is more specific. It's having more broad spectral features, more narrow spectral features. So we can identify smaller amounts of impurities. And then this is especially useful tool for looking into solution concentration. Raman spectroscopy is another tool that can be used for, especially for looking into polymorphism related issues, but it can also quantify active in the tablets, or it can be used for identification of material. So these are a few examples of spectroscopic tools that can be used in a pharmaceutical production environment. Just to give you an idea how this might look like. So this is an example of installation that I have done with my own small hands. So what you can see here is a non-contact spectroscopic measurement. So this black thing here is a lamp. So this is a radiation source. So we are illuminating the granules here in the high shear granulator. And in this high shear granulation bowl, we have a 0.5 kilo of material, and we are mixing it and then adding water during processing, and then collecting the spectral information. So what we do here is that we use this lamp for providing the radiation. So we do a non-contact reflectance measurement. So the light goes into granules, and then this optical fiber here is collecting the back reflected light. So we are not touching the material, so we can do the whole granulation cycle, and then without touching the material, keep on collecting NIR spectrum out of material. And since this all happens in a few milliseconds, then a typical granulation looks something like this. So
sorry, Jukka, there's a question in the chat. So you are discussing NIR here, but would the possibility of using IR or Raman also apply during continuous manufacturing?
Yes, indeed. Yeah, so that's a good question here. So this potentially this could be also an end-to-end continuous production line, and this would work out fine, or it could be continuous granulation process. And many of these techniques are used in the continuous processes. And I would say that it would be very difficult to do continuous process without any process analytics. So it is a key aspect of continuous processes that you measure the product quality while you are processing. So many of these commercial existing continuous production lines, for example, GEA, continuous granulation line, that is based on NIR measurements. So it is commonly used, and the same tool, same data analysis is used in there. So in principle, this could be also a continuous production line. We could have continuous material movement here, and then we would do the same NIR measurement and collect same type of information here. I'm not maybe going into details of this spectrum, but what you can see here is that I have piled up several thousand spectra during processing here. So there's a NIR region here from 1100 to 2200 nanometers. And then what we can see here is an increase in water when I added water to the granulation, and then increase in the baseline. So you can imagine that spectral baseline is kind of a shifting, tilting to the higher level, while we are increasing the granule size. So all this is kind of a visible, but it's not very clear how it is happening. And here you start needing data scientific tools and techniques like chemometrics and multivariate analysis is commonly used in this field. And what we can do here is to do a technique called principal component analysis. So what we then do is that we are projecting each spectrum here. So one spectrum becomes a spherical symbol here in this PCA space. And then we can follow the mixing of powder here and then adding granulation liquid and then the final wet massing phase here in this more simple presentation. So what we can now define here is that this is our process control window. So we always want to mix the powder until it reaches this level. Then we add water until it's reaching this level here. And then we keep on wet massing until we are somewhere here. And then that would then give us a direct measurement of degree of mixing, water content, and then granule size of our system. So this is indeed something that is of interest. And then so it is important to take this into a picture, a bigger picture there, and then think what quality by design means. So quality by design is more than the experiments and then experimental work. So then you try to understand what is happening for your product, and then this you can do both experimentally and but then also increasingly computationally. And as we discussed, so many of these techniques, they are pointing towards continuous manufacturing solutions. So in context of continuous manufacturing, these techniques would be very important. And one of the ultimate solutions would be then to aim for real-time release testing. That would be very beneficial for this environment. So as I discussed, so the regulatory framework for this exists. So if you Google this ICH and quality, so then you might find these 14 ICH quality documents. And then you can already start seeing that we discussed about Q8, pharmaceutical development, but many of these documents are also indicating the importance of continuous processing and analytical procedures for process environment. So these Q13 and Q14 are currently drafted guidelines, and then they contain the elements of continuous processing and more practical PAT considerations. Yes, but then we can here start rounding up with few thoughts on this more detailed analysis of things. So QPD is more than doing a lot of experiments. So if you think about science-based product development, so how much depth you can get on understanding of your product. So with the experimental work, you are creating a basis and foundation for your understanding. But this experimental approach gives you, so to say, statistical relationship and correlation of things. So for example, if you increase the drying temperature, then you will have less water remaining in the end product. So that is quite logical. So you find this type of mathematical correlation. So for example, increasing the propeller speed in high shake granulation, then you might get bigger granules. So you can find this type of relationship, but then you don't yet know that why things are happening. So what you should aim for doing is to gain this mechanistic understanding of system behavior. So why things are happening. So ask from yourself the question why. And then if you know these mechanisms that affect, for example, granule growth, then you can start aiming for first principles modeling. So build up a mathematical equation, rate equation for describing how this system behaves. And then this is what we do with the experimental design DOE is often empirical work. And then when we are doing for this mechanistic understanding, then we try to understand what is happening there in the, for example, in the granulation process. So in this granulation process, we have many different sub-processes happening. So we have first the dry starting material, and then by adding the granulation liquid, then we start seeing the nucleation of the first granule particles, which then start with a mechanical stress consolidating into a more condensed granules. And then by controlling all these sub-processes, then you can create even a mathematical model of your granulation. In some cases, then it's good enough to know that you experimentally know that if I add 400 grams of granulation liquid, then I get 2 big granules. And if I have 200 grams of granulation liquid, then I have 2 small granules. So 300 must be the optimum. So sometimes that is enough. But then in some cases, you would like to know more about this, that why things happen. And then you increase your process understanding. And then while doing this, then you might need to build up some more detailed analysis of the system. For example, look what is the porosity of granules during granulation, and then look into pore saturation of systems, and then how material is deforming during granulation process. And by this means, you can then build up process maps that are based on a mechanistic understanding, and then build up some decision-making rules, what you should be doing in different situations. So if you are here in a wrong part of this map, so then you can build up some rules of thumb that you should do in different situations. So if you want to increase the growth rate, then you should increase the binder content or look into the agitation, the mixing, propeller intensity, if there's something else happening in the system. Yes, so this would be a mechanistic understanding of system behavior. And then if you have, so to say, enough time and money to invest and understand the system, so then you try to build up a mathematical model. So you build up the differential equations that describe how this gas, solid, liquid system behaves. And then this can be done with the mathematical models. And here is a simplified example of that. So what we are doing here is that we have been experimentally mixing white and red spherical particles in an impeller system. So we have a propel here that is rotating and mixing the system. And you can see that after 1 to 5 revolutions, we have a homogeneous mixture. And then what we are doing in a simulation, we calculate all the particle-particle interactions, the effect of gravity, effect of this propel that is moving around, and we calculate exact position of each spherical symbol. So we can handle millions of particles in this type of system. So we can mathematically calculate how the process proceeds without doing any experimental work. But this requires quite complex and big computer power and quite complex techniques. But these are some techniques that are developing and computers are developing, so we can even describe the behavior of fluidized bed using a combination of fluid dynamics and these discrete element type of modeling approaches. So we can take many of the pharmaceutical processes and in silico with computer predict how they behave. And all this computer-based pharmaceutical sciences, that is an area that is expanding, and we are using computers for predicting the behavior of many different type of systems. So not only like a processing environment, but also looking into how products behave, so how particles are structured, and then how we can explain how particle size of a given system is affecting the process. So we can do very detailed experimental understanding of material properties, and then at the same time use different computer methods for understanding the system behavior. So I'm not planning to walk through all these simulation techniques here. So we can do molecular dynamics type of simulations, DFT simulations, and then these particle level simulations. And we can also simulate human beings. So identify different mathematical models for describing how humans behave. So the whole pharmaceutical product can be described with a computer. And then that would be of interest in the future to increase the momentum in the field of computational work and then reduce the experimental workload. So most likely you will end up doing your master thesis in one area. Yes, so you might look for, like, for example, some animal model, how you are managing to target your nanomedicine, and then what is the elimination rate of that one. So think, of course, you base up your work with experimental work, but then also look what is out there, what is possible to do with computer. So if that's possible, so then try to calculate things first, and then structure your experimental work in a better way. So what we have collected here is, in this simple illustration, is trying to contrast this experimental work with the simulation work. So we can do lots of similar experimental work in different steps of product design, and then use computers to help us in understanding this observed phenomena. And many of the mathematics and then programming is very similar. It's shocking similar to for example, molecular simulations and the particle simulations. So there are similar forces in there, similar type of computer programs are used in the interface might be looking a bit different, but the basic principles are the very same. So it is important to try to understand the basics of process simulation so that you can communicate with a different type of people. Good. And then in the very final step here, I promised to give you a final step here a while ago, but here we indeed have the final slides. So what I collected here is the terminology that is commonly used in the field. So if you read these regulatory documents, if you meet a person from industry, and so they use these expressions from ICH Q8 documents. And this is important when you are filing in marketing authorization application, a regulatory submission, and especially in the special part of that document, there's a pharmaceutical development description. So then you better use these expressions so that you are in line with the existing guidelines. So naturally, you should document quality by design QPD-based development. And then in that work, you do your risk management work properly. And important starting point for all this is documentation of your quality target product profile, QTPP. And another important expression is a critical quality attribute, CQA, that is referring to the property that is being related to this target product profile. So if your QTPP is indicating that you are making an immediate release painkiller tablet of ibuprofen, then a very likely critical quality attribute is a disintegration of your tablet. So disintegration affects the speed that you can get the drug dissolved, and then by this means the effect of the medicine. So then in this case, disintegration time would be the critical quality attribute. And then critical process parameter is something that affects that quality attribute, for example, compaction force. So these are terms that make a lot of sense. And then since it's afternoon and it's about the coffee time here, I have a cup of coffee here. So cheers for all of you. So in this beautiful cake, we have many quality properties. So for example, for this cake, critical quality attributes could be a chocolate layer thickness or moisture content of the inner cake. So this is important that you define them. So then you can build up the quality control on measurement of this thickness and then the moisture content of the inner cake. So that makes it sure that you will always get the desired quality. And when we take this to process, then we take a well-controlled machinery. So you take your oven where you can control things like temperature of different parts and then processing time. And sometimes this is all fine, but there might be also surprises. So in case of chocolate, there are sometimes this annoying feature. So you can see this effect that it starts looking a bit greasy on the surface, the chocolate layer. And this all can be solved with the installation with the Raman spectrometer. So this is simply nothing else but an unwanted recrystallization of cocoa butter. So you have managed to get a wrong polymorph out of this cocoa butter, and then the melting point of that polymorph is higher, and then all of a sudden it doesn't anymore melt in your mouth. So then you don't get the same mouth feeling of the chocolate. And then that is indeed something that you can control with a Raman spectrometer. Not maybe in a normal kitchen, but in a commercial production environment. So QPD in a nutshell, so you look at your product, design what is it that you want from it. So you want the best ever cake, and that you control by understanding the critical quality attributes and material attributes. And then you keep control of the critical process parameters while you are making it. And then this is all that you can do with the risk assessment and DOE and material science, and if needed, PAT installations. So this was a QPD of a chocolate cake, but then with a bit of imagination, we can just replace those products with tablets and a granulation process. So then you just think the same principles for the tablet. What do you want from it? Do you want a controlled release, immediate release? And then how do you control your granulation process, tableting process? And then then you use exactly the same tools. And if we take these terms to the original picture, so in the raw material properties input space, then you should use expression critical material attributes, which you then transform to critical quality attributes. So these are the product properties. And intentional variation, that is something that you can control by controlling your process. So these are the critical process parameters. And then what you need to then do is to minimize the effect of unintentional variation. So then you build up a control strategy for taking care of this unintentional, unwanted things happening in the process. So what we have been walking through today is an overview of this QPD approach. And it is important that you keep in mind that it's a generic approach for all types of pharmaceutical products. So you can make an injectable product, tablet, cream with this QPD approach. And ICH has been giving us a nice regulatory framework for doing it. And it contains very deep scientific elements in there. So you need to go in depth of these techniques and starting from risk management strategy, doing DOE approach, and then selecting the right PAT tool that is intended for the purpose. And then when possible, use computer modeling and simulation to support your process understanding and base the control strategy for all this. And then this was basically what I had in my mind for today. And I can see that it's 3 o'clock sharp. So I hope we still have a minute or two for discussion.